Good evening, everyone. My name is Connor Mao.
And I'm Jameson Cosgrove, and this is our comparative study of Nereocystis lewickiana beds in the South Puget Sound.
So Nereocystis lewickiana is a more common way of saying bull kelp. Now, if you all haven't already fallen victim to information overload, I would assume that the question you will all be having right now is why bull kelp? Why is bull kelp so important in the South Puget Sound, and why is it right now the forefront of our project? Well, we're here to answer that. So bull kelp plays a vital role in Washington's nearshore ecosystems as both a primary producer and habitat provider. As a keystone species, it supports a wide range of marine life by offering shelter, food, and breeding grounds for numerous organisms, and it also acts as a natural carbon sink, meaning that it helps to regulate oceanic carbon levels. But despite its ecological significance, it's very highly sensitive to environmental fluctuations, making it particularly vulnerable to stressors such as rising ocean temperatures. The sensitivity positions bull kelp as an important indicator species, meaning that shifts in its growth patterns, distribution, or overall health can serve as early warning signs of broader ecological changes within our South Puget Sound region.
Since the Washington State Department of Natural Resources began their aerial surveys of bull kelp in 1989, they have observed a 80% decrease in the bull kelp regions in the South Puget Sound. This is the area colored red on the map shown up there, which contrasts with the stable or growing regions in the rest of the Puget Sound and Salish Sea. The DNR attributes this to general stressors such as rising temperatures, increased commercial activity, and increased ocean acidification. Even within the South Puget Sound, however, there are more variation between individual sites, and what piqued our interest were the more site-specific stressors to get a sense for what is driving this decline.
So historically, one of the most effective ways that the Washington State Department of Natural Resources has been able to track yearly kelp bed growth is using something called aerial color infrared photography, which is to say to get an outline of the canopy coverage of the kelp forests. The image on the right here is an example of the use of aerial colored infrared photography to map the spatial extent of Day Island's bull kelp. And it's important to note that certain environmental factors, such as tides and current speed, may not be taken into account in their studying, therefore lending a degree of uncertainty to the data. So currently, the Kelp Conservation Plan is the leading initiative by the Northwest Straits Commission and the Department of Natural Resources to bring back the historical extent of kelp beds. Mainly, the current plan helps to create more marine protected areas, boost the water quality by improving land use runoff policies, and replanting the kelp by first growing it in a nursery before implanting it into the sound. However, the biggest caveat about this is that the process is very slow, recovery of bull kelp is very lengthy, and the plan is likely to extend all throughout 2025, if not longer.
For our study, we identified 3 areas of interest that the DNR has been historically tracking, those being Salmon Beach, Day Island, and Fox Island for their relative proximity within the Tacoma Narrows Strait. Despite their relative proximity, they show very different data when compared over the years to their kelp bed coverage, with Fox Island being a declining site, Salmon Beach being stable, and Day Island having grown in the past few years.
So this leads us to our first problem statement. So firstly, although Fox Island, Day Island, and Salmon Beach have all 3 been historically tracked as DNR sites regarding bull kelp beds, they have not yet been directly compared to one another or paired with each other in terms of studying potential stressors. These sites, again, were of particular interest to us, given their geographic proximity along the Narrows, really the backyard nature of where we live right now, and their differing statuses of bull kelp stabilities. We posited that as we studied Fox Island, the region will continue to exhibit a declining status, as DNR has already noted in years past. For the bed sites at Salmon Beach and Day Island, we further posited that the paired sites will yield numerically similar results in terms of potential stressors, given the closeness of the 2 regions and stable bed statuses between those 2 sites themselves.
Our second problem statement was that Titlow Beach is also a site located within the Tacoma Narrows, but it has not been historically tracked by the Department of Natural Resources, so we wanted to include it in our studies because of its marine protected area status, which we posited will have a positive impact on its bull kelp populations. So getting into our methods, Connor and I did all of the scuba diving, kayaking, and sample collection during our field work, and then all of the analysis of that data once we got back to the Marine Chem Lab. And our mentor, Mr. DeGroat, accompanied us on our dives and provided kayaks. So a huge thank you to Mr. DeGroat. As for the data we were collecting, we collected our data on 3 different variables across those 4 sites, those being the presence of northern kelp crabs, a grazer on the kelp beds, the minimum and maximum depth that the kelp beds had reached in these sites, and the available sediment for the kelp to have a foundation for.
So firstly, we'll be diving into significance of why we incorporated the aspects of northern kelp crab presence into our studies. So studying kelp crabs helps with a crucial understanding of predator-prey dynamics within bulk kelp ecosystems, as these crabs can significantly impact kelp beds themselves through grazing and mechanical damage, which we sought to further investigate by analyzing kelp crab size and growth patterns across our study sites. To structure our data effectively, we relied on transect lines as standardized sampling units. Figure 1 illustrates a DNR-created transect by Day Island's 2023 kelp bed map, which was one of the tools we used to ensure systematic field data collection at that site. It's important to note that the transect maps that were created were specifically for DNR transects, DNR sites that have been historically tracked, being Salmon Beach, Day Island, and Fox Island. And as we said, we've incorporated Tidlow into our study, although no transects have been created by the DNR for that site specifically. In terms of data collection, we conducted our surveys aboard kayaks, randomly selecting points along the transect lines. And so at each selected point, we measured the carapace width of the first surface crab encountered, so we treated each transect as an independent sampling unit, and we were able to draw variability in kelp crab size distribution across the sites. Therefore, we're able to draw a correlation between kelp crab size and the bed densities themselves.
The second variable that we looked into was the minimum and maximum depth of the kelp at each of these sites, which we did on the sites that we were able to dive at with our dive gauges, and the sites that we were not able to dive at with the boat gauge that was provided on the Mr. DeGroat's boat. And our third and final variable was the sediment, which we collected as its importance to the kelp's holdfasts, which act as its roots, which require larger sediment to be able to grasp onto and provide a solid foundation for the kelp beds. During the sites that we were not able to scuba in, we used the Van Veeb grab lowered in from the boat, and at the scuba accessible sites, the sediment samples were manually collected by hand into jars. Once we had the sediment, we took it back to the lab, evaporated the water, and passed them through a series of mesh sieves to determine the percent by mass of each sediment size category that we found.
So now diving into results and the significance of each piece of data that we collected. So in terms of what we found at each site, we'll start firstly with our findings on minimum site depths. What was surprising to us was that the mean and medium site depths, being where we observed the depth site at which bulk kelp first started to grow, was significantly deeper at Tidlow Marine Preserve compared to Day Island and Salmon Beach. You'll note in our box plots that there is no data listed for Fox Island, being that as observed by the Department of Natural Resources in 2023, they noted only 2 bulk kelp plants in that region, and when we conducted our dive surveys at that site, unfortunately no bulk kelp plants were to be found in that region. So in terms of statistics, we decided to use an analysis of variance test, which is a method used to determine whether there are significant differences between the means of 3 or more groups. And through doing that, we used the coding language R, which is a statistical computer science language model. So because an ANOVA revealed a p-value of less than 0.01, we are then 95% confident that there is indeed statistical significance present between the different minimum site depths of each of these 3 noted sites. So based on our data for maximum site depth, it was very interesting to us that Day Island and Salmon Beach continue to hold similar results in terms of their depth distribution by our methods of scuba and kayak monitoring. Additionally, depth range of bulk kelp seemed to be largest at the Tidlow Marine Preserve. So another ANOVA test revealed another p-value of less than 0.001, and then again, we are 95% confident that there is also statistical significance in the differences of maximum site depths of the bulk kelp beds at each of these 3 sites. So now focusing into our statistical results based on our findings with kelp crab populations. So at only 2 sites, there were observed surface kelp crabs present at only Titlow Marine Preserve and Day Island as expressed by our box plots. Fox Island was found not to hold any kelp crabs, being there were no bull kelp plants to exist to begin with, and Salmon Beach was found not to hold any surface kelp crabs when we conducted our surveys. Using a paired t-test, which accomplishes the same goal as an ANOVA, but really only examines the means of 2 groups of data, we once again confirmed that there is also statistical significance present between the mean crab carapace widths of Day Island and Titlow Marine Preserve, being that the mean carapace width of kelp crabs at Day Island exceeded that of Titlow.
And as for the results of our third and final variable, the substrate, what we found were that Day Island and Salmon Beach exhibited much coarser, larger grains, whereas Titlow and Fox Island had much smaller, finer sediment found at the seafloor there.
So going into discussion and what really our results hold for the significance of the bull kelp beds themselves. So the variables we monitored, once again, were the presence of kelp crabs and the carapace widths, the bed depth ranges, and the sediment composition, which we believe are likely to be related to recent bull kelp trends with its health in a current crisis as it has already been for Fox Island. So firstly, the larger kelp crabs at Day Island are a very likely indication of a healthy bed able to support a wider range of marine organisms, with differences against Titlow being statistically significant. This leads us to believe that Day Island is a more suitable habitat for grazers like kelp crabs to settle, although further investigations on whether larger kelp crabs are inherently bad for the beds themselves should be investigated. Additionally, Titlow's wider depth range points to a greater resilience of bull kelp to more extreme environmental conditions. This could indicate that the environmental conditions at the Titlow Marine Preserve, such as water clarity, nutrient availability, or reduced human disturbance, are more favorable for deeper kelp growths at the Titlow Marine Preserve.
And finally, the larger sediment sizes found at Day Island and Salmon Beach correlate with the healthier kelp beds found there, while the lack of suitable anchoring spots at Fox Island could have contributed to the decline of the kelp found there. The overall smaller sediment found at Titlow should be noted. However, there remains multiple larger rocks and pilings that the kelp is able to hold on to and provide a foundation for that bed. That was not accounted in our data. Getting into some of the limitations that we found throughout our study, differing data collection methods were employed. Some of these sites, as we mentioned, were not scuba accessible, so we had to take the boat, which opened up to different methods of our data collection. However, they should have received the same results. We also encountered day-to-day variation at our sites. None of this was done on the exact same day, so tides and weather could have impacted the results as well. And then, of course, the lack of established transects and prior data from the DNR at Titlow Beach, which could have affected our surveying sizes without a pre-mapped extent of the kelp beds there.
So there's many potential future avenues of research that we highly encourage. One important area of study that we highly encourage would be the investigation of the relationship between bull kelp and other species of kelp that can compete for similar resources, as really this competition can affect the overall health and density of kelp forests. Also, we hope that an analysis of the kelp density at Salmon Beach could provide some more valuable insights into why exactly there's an absence of kelp crabs in that area. To us, it was really interesting that despite the presence of bull kelp, there were no kelp crab grazers to be found in that area. However, this observation is really nothing new to the DNR, as in years past, they have historically noted a near absence of kelp crabs in that area, although the exact causes are not well known. Additionally, we hope that there would be continued studies of the aerial monitoring at Titlow Marine Preserve, which could provide a broader perspective on the true spatial extent of the kelp forests, which would allow for more efficient tracking of the sites, just so that it can come under the umbrella of the DNR historically tracked bed sites. Finally, we also hope for a more comparative studies of other factors at Fox Island, Day Island, and Salmon Beach, factors such as ocean acidification, pollution, or fluctuating water temperatures, the purpose of which would be to understand how these other stressors could predict the future of bull kelp populations, which again are highly sensitive to environmental fluctuations. So in conclusions, our results bared some notable observations. In the end, our first hypothesis was supported in that Day Island and Salmon Beach yielded numerically similar data regarding the variables surrounding bull kelp environments.
And then in the end, our second hypothesis was inconclusive, as the marine protected area status at Titlow the data we found there did not correlate enough with the healthier sites found in the rest of the Tacoma Narrows. A huge thank you to Mr. DeGroat, our mentor, for all of his support, Ms. Harrison for giving us access to Salmon Beach, our parents, of course, thanks Mom and Dad, and then the Washington DNR and Reef Check for providing us with the historical data, and of course the entire Marine Chem class of 2025. Thank you all.
Yeah. And so now we'll be opening up the floor to some questions. Yes, David.
Yeah, so if the sediment at Fox Island can't support kelp, then how can the kelp bed ever form?
So the question was, if the sediment we found at Fox Island is not large enough to support kelp, possibly, how would there have been a historical bed there? And we looked into this actually, and Fox Island is the only site that has historical geoduck harvesting found there, which is interesting for those of you who don't know. Geoducks are clams found out in the same depth range as the kelp, and the harvesting process for that is they send down divers with high pressure hoses to kick up the sand, uncover the geoducks so that they can collect them, which would cause the finer sediment to rise to the top and disturb the sediment found there. Any other questions? Sol.
You mentioned about hoping there be further analysis on the absence of shelf crabs. Do you have any current hypotheses regarding predation or disease for the absence?
Absolutely. The question was, to our knowledge, could there be some other factors as to why there is an absence of kelp crabs at the Salmon Beach sites, which was interesting to us. One notable observation that we saw at the Salmon Beach sites was that the kelp bed was tightly compacted and its density was much higher than that of Day Island, meaning that the kelp would practically grow on top of each other, leaving less room for a sunlight attenuation to reach the ground floor. So we possibly point to this as the overbearing density of Salmon Beach to be one of the factors that possibly kelp crabs are not able to settle properly on the beds. As with Day Island and Titlow, the kelp beds themselves were more spaced out and across a wider range, while the kelp beds at Salmon Beach were more linear and more compact. Although the exact causes are not well known, we'd love to investigate that more.
Sabe?
I was wondering if you've ever been curious to contact previous marine chem students on account of data of aerial photography around Titlow Beach.
So the question was whether we had contacted any past marine chem students, possibly Salve's brother, who did an aerial study of eelgrass at Titlow. We were really focused on the bull kelp there and not the eelgrass, so we did not reach out.
You guys touch upon efforts to bring the bull kelp back to restore it?
The question was, did we touch upon efforts to bring back the kelp? Yes. The answer is yes. The kelp Puget Sound Kelp Recovery Plan is the plan that we mentioned early on, where they're imposing economic and environmental restrictions to protect the kelp, as well as seeding nurseries to release start the kelp out, and then they release that into the wild to get it started. Anyone else? All right, thank you all.